Hi, I'm Steve Palumbi, and this is the Extreme Life of the Sea. This lecture today is about the way natural selection works on populations in extreme environments. We've covered environments like the hottest ones in the ocean, the coldest, um, and the organisms that live there are adapted to those places. But what's the process by which natural selection adapts species into their environmental niche? And that's what we're going to talk about today, the way that selection acts on the genomes of a diverse kind of marine species. First, we delve into the nature of genetic variation in a species. A lot of the genetic variants that we find in marine species, and there's a lot of them in marine species, a lot of those variants really don't play a big role in adapting the species to their environment because selection isn't acting on them at all. They're called neutral genetic variants. And over the past couple of decades, a set of theories about how natural selection works on genes has changed our view of what we should find in the genomes of organisms in their natural environments. If you sort of take a look at the selection theory, that is, genes are being controlled by selection, then the variance you see in a population, that theory suggests that a lot of those are deleterious because random genetic variation is likely to be deleterious, and there's some that are advantageous. But over the last couple of decades, the neutral theory of evolutionary change has really been seen to be more likely to fit with our observations of genetic variation in species. And that posits that, in fact, a large fraction of the existing genetic variation that's out there is neutral. It's not being acted upon by natural selection. There's still a good chunk of those that are deleterious. These are new mutations that just have arisen. Selection is getting rid of them. And the theory basically says, well, since most genetic variants are deleterious, selection will get rid of them. The ones that are advantageous, this little corner here, if they're really advantageous everywhere, then selection will make them fix everywhere. They'll essentially be so common as to be ubiquitous everywhere. And the ones that are still polymorphic are the ones that selection is neither decreasing because they're deleterious or increasing because they're advantageous. And these neutral genes then, it turns out, make up the vast bulk of the genetic variation that we see in most species, even in extreme environments. But over decades, people have been exploring genetic variation and have discovered some that are playing a strong role in adaptation. And those examples teach us a lot about the way natural selection works, and in particular, what maintains those polymorphisms in populations. So one example is this one from the east coast of the United States. This is a marsh minnow called Fungulus heteroclitus, or the mummy chug. And on the x-axis here is the position along the east coast from northern marshes down to warmer southern ones, Georgia and Florida down here, Maine, Newfoundland, and up north up here. And in northern environments, you see one particular allele, one particular genetic type of a protein called lactase dehydrogenase. And that protein, or that allele, is the B allele at this particular gene, and that particular allele is practically fixed in northern populations. But if you go down to southern populations, that allele is practically absent. And in between is a shift in the allele frequencies of this particular gene up and down the coast in a gradual way, and this is called a genetic cline. And you see that, for example, there's about a 50-50 mix here in mid-latitudes. This happens to be the marshes of New Jersey. Well, this shift in the genes from north to south can be explained in a number of different ways. It could be that this is just a natural random event, but in fact, the biochemistry of this particular allele, of this particular protein, shows that it works better at colder temperatures, the B allele does, and the alternative allele, the A allele at the same locus, works better at warmer temperatures. And so by taking this genetic cline and then looking at the physiology and the action of the actual proteins that the genes produced, it's possible to mesh the actual function of the genes with the genetics and show that this cline is actually constructed and maintained by natural selection for the B allele in the north and against the B allele in the south. And that's the balancing act that often we see for populations that have these polymorphisms, that each of the alleles that make up the polymorphism is in fact better at at its job at one place or one time or one condition or other compared to another allele. Well, another example also from the East Coast shows a very similar sort of dynamic. This is a mussel in the genus Middleus. This is Middleus trocylus. And the population studied here is in Long Island Sound along the East Coast. And what is shown in this graph are the allele frequencies of a particular gene, not lactate dehydrogenase like the previous example. This is leucine aminopeptidase. And the 94 allele shown in red here is pretty common out in open ocean settings. But as you walk into Long Island Sound and you get further and further and further deeper into the sound, then the frequency of that particular 94 allele goes from about 50-50 down to only about 10 or 15% in the population. So there's a strong cline, just like we saw in lactate dehydrogenase. It's graphed differently here, but it's a cline nevertheless. And it's also due to natural selection because the researchers who studied this particular cline were able to show that the different alleles affected the ability of small mussels when they first settle in and land to be able to grow and survive winter conditions, and the allele that they need to survive and grow in winter conditions way down in the sound is not the 94 allele. That's selected against there. A little bit differently than the Fungulus example, these researchers were also able to show that every summer that reproduction of the mussels up in this part of the sound then allows a huge cloud of larvae to come down into this part of the sound and settle and start to grow. They bring the genetic signature of the open ocean setting with them. They bring the wrong alleles with them, and then every winter you can see natural selection working to winnow out the 94 allele and reduce it in the population. So in this case, we have the geography, we have the biochemistry about these alleles, and then we also see the temporal dynamics of natural selection working on these. Well, these examples have become pretty well known because they're rare, because they don't occur in every species in every circumstance. And in fact, if you just look at a few genes in marine species, the chance that you're going to find some under selection is relatively low. Those studies were done when only a dozen genes or so were able to be looked at in the genomes of organisms. As time has gone on, the number of genes that could be looked at has gone up and up and up. Those were all protein studies. Genetic markers called microsatellites can use tens of different alleles or different loci, 10, 20, 30 is pretty common. There's ways of looking across the genome randomly for thousands of different loci to see how genes interact. And then if you look at transcriptomes and then genomes, you have access to 20,000 or so different genes. If you look at genomes, you might have millions of polymorphic bases to look at. So as the power of genetics has grown, so as the number of different genetic loci that people can look at, well, eventually you get to the point where you have so many loci that it's pretty likely that selection will be acting on some of them. And by looking across large sets of loci, it's been possible to find the signature of selection in many different places than we had been able to before. The reason why this is important is that the different loci are acted on differently, and you can see that in their features. So something called genetic drift is a feature that it affects all loci in a set of organisms exactly the same. That's essentially the random success or failure of individuals in the population, not depending upon their genes, but just depending upon chance events in the population over time. And so as a consequence, genetic drift causes gene frequencies to change little by little year after year in all populations. Big populations, they don't change so much. Small populations, they change a bunch. The important thing is that it happens across virtually all loci the same. Dispersal also brings all loci along with it in a pretty even way, but natural selection works just on some loci and not others. And so the ability to find loci under natural selection depends upon looking at how loci are differing from one another over space and time compared to what we expect to happen from genetic drift and dispersal. And by looking at those signatures across thousands of genes or millions of single nucleotide polymorphisms, that's how the signature of selection is able to be found in populations using these large amounts of data. Well, I want to give you an example from our own work on corals in the Pacific, and it also ties up into the way different very similar species are differentially adapted to different conditions. In this case, different conditions of warm water temperatures on reefs in the Pacific. What's shown here is a set of corals along this column here that we've called species A, C, D, and E. There is a B, by the way. It's just not on this graph. And these are very, very similar corals in a species complex that's called Acropora hyacinthus. It's a kind of a tabletop coral that grows like this big or even bigger on reefs all over the tropical Pacific. And then looking at each individual coral, they can look very, very, very similar to one another, but the underlying genetics told us years ago that there were, in fact, several species here called cryptic species. You couldn't really tell apart physically very easily, but you could tell them apart genetically very easily. And they've been called A, B, C, D, E, and F. They're distributed around the Pacific in this sort of way. Where we worked in Samoa, there's 3 of this hyacinthus species complex, for example. In Palmyra, in the middle of the Pacific, there's just 1 species F. Palau, where we're also working now, there's also 3, but a different 3 than are in Samoa. So we know there are genetic differences among these corals. They fall into good species. And how are they different from one another? And are those differences, do they have anything to do with the environment that these species live in? And it turns out that in Samoa, where we have done this particular work, that one species in particular, species E, is a lot more resistant to high water temperatures than the closely related and almost indistinguishable other cryptic species that is in Samoa, so species A and C and D. Shown here are differences between species E, the heat-resistant one, and species C, which is the most common of the other ones, in 2 different locations. This is a pool, the highly variable pool, back reef pool that has got a lot more warm water conditions. This is a moderately variable pool, much more cooler water conditions. And where the water is cooler, these species E and C are relatively similar in their abundance. Where the water is a lot warmer, then species E hugely dominates these particular areas. So a big difference in the species depending upon the water temperature that they're found in. This wasn't at all obvious until we knew the genetics underlying it, because these things look the same. So we had no way of telling. We can also then look at how each individual reacts to specific kinds of heat experiments. This graph is a set of individuals that are in their natural environment, and we suspect that this was due because of heat but there could have been other things that are different between these pools that drove this change. In this particular case, we've taken these corals, we have tested them at 33.5 degrees, at 34 and 34.5 degrees to see how they react. And what's shown on the y-axis here is the amount of chlorophyll that they retain when you heat them in these conditions. The amount of chlorophyll that's retained in these conditions is an index of how heat resistant they are. If they retain all their chlorophyll, they're very heat resistant. And what you can see is that the species E retains more chlorophyll than species C at 33.5 degrees and at 34 degrees, and even at 34.5 degrees, although the numbers are all low here. So in the natural environment and in the lab, species E is more heat resistant than the other cryptic species. So we know there's a physiological difference between them now. We know there's a genetic difference between them now. And the question is, how do those two things come together? How does the genetics and the physiology mesh? We can also see that those differences are important when bleaching events happen. So these are results from the 2015 El Nino, the bleaching events that came across American Samoa that time, causing corals to turn white, bleach, and die because of high water temperatures there. And what you can see here is that species E had a very small fraction of its individuals that visibly bleached. That's just this much compared to that much. Whereas the other species, the other cryptic species in this same species complex, had a much higher fraction of their colonies visibly bleached this same particular time. So what we saw in the natural environment, what we saw in the experiments is also seen when we have these extreme events, these bleaching events that come sweeping through. A graduate student in my lab, Noah Rose, led the study of a set of individuals of these species by taking 114 colonies, doing entire genome sequencing of all 114, and then looking to see how different each individual colony was depending upon what cryptic species it was. And what Noah did was to draw each cryptic species with this little diagram that showed the genetic difference between the species and all the other ones. The basic diagram, though, just looks like across all the species are about equally distant from one another when you looked at the genetics of these individuals. So HE, that has a very different heat resistance, isn't standing out here. But if you take genes that we thought from earlier work were involved in the heat reaction, the heat resistance reaction, and you sort of make a list of those genes, and there's a list of about 1,000 of them based upon transcriptome work and other work we've done moving colonies around in Samoa, take that list and say, how do these different species differ from one another at that list? Then we get a very different picture. Then HE becomes much more differentiated from the rest at this set of genes that we had an idea were associated with heat resistance. So the heat resistance gene list is much more differentiated in this heat resistant species than the average gene. And that corresponds to what we would think in the neutral theory would suggest that most of these genes are just affected or not under selection whatsoever, and so they're all about equally divergent within these four species. But if we pick the list that might be under selection and look at those, then we see this other pattern of higher levels of differentiation in HE compared to the rest. So then we go to the next stage and say, well, across the genomes of these species, then how do they differ? How do individuals differ from one species to the other? And what this graph shows across about 330 million bases of coral genome, it shows the differences between individuals of species A and the rest of the species, C, D, and E to the rest of the species. And then the colors here are that the sort of the average that we see across all genes is in gray, and genes that stand out a little bit, genes that look to be a little bit more divergent at any particular part of the genome are colored and stacked up. So your sort of eye can go across there, and what you can see immediately is that there's some places where every species has a standout, has a place in the genome that is different quite a bit different from the other three. So this area in HC, this area in HA, and we concentrated a lot on what the differences were in HE because we were interested in perhaps what the genetic control of that, the one trait we knew was different in HE, and that is heat resistance. So we concentrated on two areas that are called SL1 and SL2 on this particular graph. They're the standout places that look to be differentiated between HE and the rest of the species. And then from this graph, we can dive in, see it a little better here, and go further in to say, well, what are the genes that are in this region of these particular parts of the chromosome, and then what might they be doing in causing or helping species HE to be more heat resistant. Well, the region SL1 is a pretty small region. It's only about 174 kilobases and has about 7 genes within it. It's highly divergent in HE. It's a little bit polymorphic, but it's got the alleles in HE are very different than the alleles in that same 174 kilobase region in the other species. SL2 is an even smaller region. It's 74 kilobases. It has 5 genes. It's completely fixed in HE. There isn't any genetic variation at all in the warm water resistant species compared with the other corals. We dig down even further and say, well, no, we have a piece of the genome that seems to be under selection. It seems to be very divergent in this species with high heat resistance. What sort of genes are in there? So for SL1, there are 7 genes. These are their lists here, including 2 ribosomal proteins, a transcription factor, 3 proteins we can't characterize, and then 1 that is involved in ubiquitination of proteins as they're being degraded. We can see 2 things about these genes. The data are not shown here, but we can see that the changes that happen between these genes and the other species, there are more amino acid changes in these proteins than you'd expect by chance alone, suggesting that selection is acting to change the protein sequence in these genes in HE compared to the rest of the species. But the other thing that we can see is that the expression levels of these genes are also different. What's shown in this graph is the expression level of this gene, the TMF, the tide element modulation factor, which is relatively low for the alleles that you see in species A and C and D. But for the allele that you see in species E, that's over here, the expression levels are much higher. So not only are there amino acid changes between these proteins, but they're expressed at different levels, and both of those could be part of the reason why selection is acting on this region in regard to their high heat resistance. Now, honestly, we don't know exactly the reason why these changes might be associated with high heat resistance, and we're trying to find out more about it. But by taking millions of bases of coral, looking to see which ones are the standout of the species that has high heat resistance, and then focusing on these gene regions, we're beginning to narrow in on some of the possible causes for heat resistance in these species. I should say that all along in this research, we've found not just 1 or 2 genes, but we found potentially hundreds of genes that are involved in heat resistance within species. And that we also see for these corals that you can move them around from environment to environment. And if you move them into a warmer environment, they will acclimate a little bit. They will gain more heat resistance through living in a warmer water environment. So from the standpoint of the corals themselves, their ability to resist heat is made up of 2 facets. One is their acclimation, how much time they have spent in warm water in the past and how that's changed their physiology. And the other is their intrinsic genetics, like at the SL1 or SL2 loci. Together, the acclimation and the physiological adjustment and the genetics can give you a wide range in phenotypes in coral heat resistance, which is what we see out in the field. So why is this at all useful? Why is it important to know the genetics of something like heat resistance in corals? And the answer comes in a couple of different categories. One is predictions for future climates. Second is tools for coral restoration. And then after that is the global search for heat resistant corals around the world. Key set of tools depends upon us knowing how the mechanism of the genetics of the corals then ends up affecting their phenotype, their heat resistance phenotype. Because for corals, there's in fact not just two reasons for them to have heat resistance, but there's potentially four. I've been talking about the genetics of the colony itself. I was talking about their acclimation to different environments, but there's also the symbionts that live within them, because those symbionts can come in different genera of the small single-celled algae that live within them, and some of those genera are more heat tolerant than others. And also the microbes that live among the corals. So microbes and symbionts and acclimation and genetics are all figuring in to how the corals are actually heat resistant. But the microbes and the symbionts and the acclimation are all temporary. They change over the course of the coral's lifetime. They change with their environment, whereas the genetics doesn't. And if we really want to use heat resistant corals in the future and understand how they might evolve, then we have to be looking at the permanent part of their adaptation, that is the genetic part. So a lot of the work that's gone into this sort of field has been in looking to see what variation there is in heat resistance and then pulling apart the reasons to be able to focus in on the genetic part. To look at the question about how corals might be able to evolve in the future and how the genetic mechanisms that we understand can help that along, we can take a look at the Cook Islands in the South Pacific. Cook Islands are closer to New Zealand and Australia than they are to the United States. They're also so far south here in the South Pacific that they're some of the most southern populations of highly diverse reef-building corals in the Pacific. The cool water that they see in the winter is actually a little bit lower than their normal population ideal temperature. And so it's conceivable that as ocean temperatures rise, that these populations might actually be getting into better water conditions than they had been before, rather than worse water conditions. So the question really is, if these corals are adapted to cool water, what's the chance that they can evolve adaptations to warmer water over the next couple of decades as ocean temperatures rise? Well, that's in particularly answerable in our case because we also have a great deal of data on the genetics of these corals from an archipelago that's close by, that is Samoa, which is about here on this map. So we can take our data from Samoa, particularly from the very warm water conditions that we find in some reefs in Samoa, and ask, what are the alleles of those genes that we see in the Cook Islands? And can we imagine that those alleles will evolve to be more and more frequent? And that is, the warm water alleles will evolve higher and higher and higher frequencies over time. What are the limits to that? So we started with coral populations around the western side of Rarotonga, sampled those, sequenced them genetically, and found out whether or not they had the same alleles for warm water adaptation than we found in American Samoa. And the answer was that they did. Those alleles were there. They were just at a much lower frequency in Rarotonga than they were in Samoa. Well, now that we knew that they were there, we could begin to ask the questions, under what circumstances could we imagine those frequencies of warm water alleles could go up over time? And could the evolutionary process keep up with what we expect the warm water change? Can they keep up with what we expect temperatures to do during ocean warming? So we went back to projections about future ocean conditions in different parts of the world. And that's what's shown in this graph here from 2015 up to the end of the 21st century. We can see the expected temperature rise across here, but we've also can see it with different types of CO2 emission scenarios. They're called RCP scenarios. RCP 2.6 is this dark blue one. Essentially, we've already exceeded that. We've all but already exceeded 2.6. The next one is 4.5, which we could basically hit if the Paris Accords are followed. That is the promises that nations have made to reduce CO2 emissions around the world. If those are followed, we might be close to about RCP 4.5. If we don't follow any of those and countries continue to emit CO2 into the environment at the rate that they're doing now, then that's RCP 8.5, and that's the projected increase in ocean water temperatures around Rarotonga under those particular conditions. Well, that tells us what the ocean conditions are likely to be like in the future. Then we can take the alleles that we know are there from our work in Samoa and Rarotonga and ask with a mathematical model, if the temperature goes up, how much will that cause selection on the population to increase those allele frequencies? If the temperature goes up too fast, it's likely that too many of the corals won't have the right alleles and they'll die. If water temperature goes up slowly enough, then the process of natural selection might allow corals to basically be able to breed generation after generation, higher and higher and higher frequencies of these warm water alleles. So a study done by a former student of mine, Rachel Bay, simulated the evolution of these colonies of corals in Rarotonga under these different types of heat scenarios. And what's shown here is the estimated population size of corals in Rarotonga, basically with a maximum of 1,000. So this is just a small area of a reef. Over time, under the different heating scenarios, allowing this evolutionary mechanism to work. So what you can see for the dark blue line for RCP 2.6, that water temperatures are not expected to go up very much, and populations are pretty fine all along the way. Ironically, this collapse here at the end is not because temperatures go up, but because of a strange part of this temperature models. Temperatures right at the end of the century are projected to drop here in this model. So that's what's causing the drop here in this part of the prediction. If we go to RCP 4.5, a little bit more quick increase in temperature over time, that's shown here, then the coral populations basically do okay for a while, but then they drop off seriously. That's because of the impact of increasing water temperatures on their ability to survive. And then they begin to evolve and then come back up. And so in this particular scenario, the process of rising temperatures has a negative impact on the corals, but evolution basically turns that around and allows the population to evolve to be adapted to those new temperatures by the end of the century. For RCP 6, that is kind of halfway between 4.5 and then the business as usual strategy in red. For that strategy, what we see is a decline in coral populations. They begin to evolve to come back and be more and more heat resistant, but the water temperature is moving too fast, is increasing too fast, and the coral evolutionary rate can't keep up. They drop off and go extinct, but not until the latter half of the 21st century. Under business as usual scenarios, this red one, the coral populations decline, they go extinct, and they can't recover. So this is just one of a number of models that have been published lately that tries to look at the balance between selection, natural selection because of a change in environment, and genetic polymorphisms in coral populations, and understanding the way those genetic variants work and how they affect heat resistance is really fundamental to making these models work. In this particular model, we made some generalized assumptions about how they work, and then future research really should focus on whether those assumptions are really the right ones. So this is a prediction for the future, and it also suggests that populations might actually be able to evolve to take advantage, or it suggests that populations can evolve in the future to adjust to climate change if that climate change is not too fast. But other people have been thinking more in more short-term cases of saying, well, what else can be done to try to bolster coral reef populations? Is there any way to use heat-resistant colonies in order to, for example, make current reefs more heat resistant than they would otherwise? This evolutionary process will play out over decades. Is there any way to accelerate that? So question is it possible to use heat-resistant corals to build coral nurseries that are themselves more heat-resistant in the future? Coral restoration has become more and more an important part of what people are doing around the world to bring coral reefs back after they have declined because of sedimentation and overfishing and climate change. But you can imagine that there's a bunch of different coral restoration strategies, one of which is just to restore whatever corals are there. The other is to say, well, let's find the ones that are the most heat resistant and focus in on those. But does that work? Does focusing in and choosing heat resistant corals to make coral nurseries actually give you a more heat resistant nursery? Because remember, there's multiple ways that corals attain heat resistance. There's the symbiont, the microbes, the acclimation, all of which are temporary, and the genetics. How much is the genetics of a heat nursery or a heat-resistant nursery going to determine the heat resistance of those corals? So in this particular experiment, graduate student of mine, Megan Morikawa, laid out nurseries with a variety of different corals that were either heat resistant or not heat resistant. They were taken from those two areas in American Samoa, the warmer pool and the less warm pool. And so for 4 species, she mixed together heat resistant and not heat resistant colonies to see how they would grow and how they would thrive in these mixed nurseries. What she found was that they grew about the same, but in the 2015 bleaching event, which came across American Samoa, these particular coral nurseries did very differently depending upon where the corals were from. And what Megan has done here is circle branches and nubbins of corals that came from cool water habitats, and they were not heat resistant. That's the ones in the blue. And circled in red are the corals that were heat resistant that came from the warm water part of the reef. What's shown here is the results after the bleaching events that happened in American Samoa in 2015 and 2016. And what you can see is that the corals that came from the cooler water parts of the reef bleached a lot more than these other corals. In fact, bleaching in the cool water corals was 2 to 3 times higher than bleaching in the warm water adapted corals. Well, these corals have been growing in this part of the reef in these artificial nurseries for about 1.5 years. So whatever acclimation they had was gone. Whatever microbes they had the chance to shift, and the symbionts also had a chance to shift. So the other mechanisms that could have caused these differences in the coral reaction to heat were basically uniformized in the nursery, and the only thing difference between them is the genetics of the colony. And we can see that, in fact, making nurseries with these heat-resistant colonies creates a heat-resistant set of corals in these nurseries for the future. So that allows the possibility that coral restoration could use these colonies and then grow more heat-resistant reefs in future conditions. That was a lot about corals and a particular kind of adaptation. And we were talking about the genetics of these organisms from the standpoint of a lot of different genes scattered across the genome. But it turns out that there is another feature of the adaptation of organisms to different environmental conditions that's not just about the genes themselves, but how they're organized in the genome. The architecture of these genes in the genome is an important part of how species seem to be adapting to current variation conditions and in potential future conditions. An example of that is data from Carolyn Teppold's work on green crabs as they had invaded North America. Green crabs are very common along the coast of Europe. From northern Africa all the way up into Norway. They invaded the east coast of the United States in two waves, one in about 1817 into the central part of the United States, and then another invasion, another transportation from northern Europe into the northern east coast of the US in the 1980s. And then in the late 1980s, there was another invasion from the East Coast into San Francisco Bay, and they subsequently spread north. So in this particular species, we've got an original range, and we've got a range of the species after it had been transplanted to North America. This range has a wide set of temperatures. This range has a wide set of temperatures as well, but the invasions came from just two spots. The question was, well, has this population adapted to its range of temperatures compared to this population? What Carolyn was able to show by looking at single nucleotide polymorphisms was that she could take the data, these are polymorphic parts of the genome, these are genetic variants, and she could divide them up under these variants that seem to be neutral. That is, they seem to be following the simple rules of genetic drift in populations. And she could actually distinguish the North American population from the European population, and then the Newfoundland population, which was basically came from Europe in the 1980s. But when you looked at SNPs that are under natural selection, then the difference between those populations begins to pull apart. The northern and southern European populations pull apart like that. The populations within the east coast of North America pull apart. This is a blow up. C is a blow up of this box here. And the SNPs that are under selection are more differentiated on the east coast of the United States than the neutral SNP. So it gives us some evidence that, in fact, selection is working on these genes to reestablish the kind of genetic line seen in Europe, to reestablish that in North America. Well, Carolyn could do physiological experiments on these crabs and rate each population in its tolerance to cold. And that's one of the biggest features of the European population from Norway down to northern Africa. And she could, for example, graph the genetics of one particular gene. This is a ryanodine receptor. Changes its gene frequency depending upon the cold tolerance of the population that's involved. So in this case, there's a link between this gene, the ryanodine receptor, and the cold tolerance of this population. Here's another example: a different protein, a structural protein. Showing the same pattern between cold tolerance and the alleles. Well, as Carolyn was digging through the genetics of this crab population on the East Coast that was just reestablishing its genetic lines, what she found essentially were 50 different genes that looked like this and this. And that's what's shown on this left-hand graph, is a superimposition of the data from 50 different genes. Well, the 50 different genes then are involved in the adaptation that we see in the crabs as they have moved up and down the east coast of the United States. And it looks like the adaptation to cold water is not just one gene, but it's a whole series of genes. Well, that's probably what's happening in corals too. There's a whole series of genes that allow heat tolerance to evolve. But in the case of the crabs, these patterns are just so easily superimposed upon one another that they actually look like this. It actually looks like these 50 genes are actually acting like just one gene instead. So to look at that, what Carolyn did was to look at the correlation between the genotypes that crabs have at a bunch of different genes. Essentially, she was looking at what's called the linkage disequilibrium between these different genes. Think about it a different way. If I tell you that my genotype at the ryanodine receptor is an AB, it really shouldn't tell you anything about what my genotype is at this other gene. But if these genes are linked, and I've got an AB genotype at this gene, then if they're really highly linked, I'm much more likely to have an AB genotype at this gene too. So it's the ability of the genotype at one gene to predict the genotype at another gene. That's essentially what high linkage is. Well, when we look at that high linkage, we can see it in a number of different ways. So here on this graph, shown down in these row after row after row are different individuals. And the I'm sorry, the row after row are different genes. The columns are different individuals. These are North American individuals. These are European individuals. And when you see columns of the same color, it means that these individuals across there all have the same genotype across many different loci. So the fact that these columns are so easily discerned means that the genotype of an individual at one locus tells us almost perfectly what the genotype is at another locus in another individual. Well, we take that and ask what is the correlation between the genotypes of one locus versus another. We do that pairwise, and we get a graph that looks like this. Red means that those are highly related genes. They're highly linked to one another. That is, the genotype of one has a high predictive value about the genotype of the other. As the graph gets to be more yellow and green, that correlation drops off, the linkage drops off. What it means is that this is a highly linked block of genes that is related to one another, but it's also highly related to the temperature that these organisms were found in. So what it looks like here is that there's a block of genes that allows for temperature tolerance in these crabs. The genes themselves may be in one part of the chromosome. They may be physically linked to one another, and so get passed on to offspring all as a group. We don't know that for this particular species because we don't have a full genome, but it's probably the case that they're all physically as well as genetically linked. When people have talked about the linkage among genes in genomes and on chromosomes and their role in adaptation, they often call them supergenes. It is sets of chromosomes that have adaptive genes in one section of those chromosomes, and that the fitness of the organism or the adaptation of that organism to that environment is the sum total of all of those genes in that section that contributes to that adaptation. And we've seen that kind of organization in the genome in a bunch of different places, like in different color morphs of butterflies, in the lengths of stamens in these particular plants. By having a set of genes that are going to work together to produce a particular adaptation, then those supergenes can be transmitted to offspring in one unit, makes that transmission, makes that inheritance of that adaptation a little bit more direct and a little simpler. So we saw some of that in the example of the green crab, although we don't have that much knowledge about the genome of the green crabs. And there are other examples, though, among marine organisms that seem to be pointing in that direction. And in one case, we actually have some experimental evidence of that. And that comes from a small minnow here called a silversides. And these minnows were the subject of a set of experiments done by David Conover and Steve Munch and their colleagues a couple of decades ago, the late 1990s. What they did was that they had a population of silversides, which is that are individuals about that big, and they pretended to fish them pretty heavily in experimental populations. And in one set of the lines, they took out all the small individuals. In another set, they took out all the large individuals, like fishing would take out the large individuals out of a fished population. And what they saw over only 5 generations was that the populations evolved to be bigger when you selected against the small ones, and they evolved to be smaller when you took out all the big ones. And that's it corresponds to what people have seen in natural wild populations of overfished species. They have tended to evolve to be smaller and more slowly growing. So in this experiment, what they had was 2 up lines, that is 2 lines that were selected to be bigger, 2 down lines, 2 lines that were selected to be smaller, and then 2 control lines that were selected to be the same size. And the original papers were about the response of these lines morphologically. They got bigger and smaller, and in terms of growth rate. But all of these fish were also saved. They were preserved. And then 2 decades later, Nina Thurkelson, who was working in my lab at the time, was able to use those to look at the genetic changes that happened over 5 generations of strong selection in these fish. And what she found was that the genetic changes that occurred in the up lines and the down lines occurred over thousands of genes, which was expected because of the strong influence of strong selection on them, but that there were other parts of the chromosomes that seemed to be hotspots of adaptation during this evolutionary process. What's shown here are the changes that occurred in these minnows across all 24 chromosomes in the up lines, the control lines, and the down lines relative to one another. And if you sort of scan this set of data, for example, you see that, okay, chromosome 9 has a large number of loci that are strongly differentiated in this particular experiment. We also see strong differentiation in chromosome 12 and chromosome 15. Sometimes different lines have different sorts of results. For example, the big bump in 9 here really isn't seen in the up the line or up to. But there's a couple of them that really stand out. And in one in particular, chromosome 24, in one of the down lines, there's this incredible big burst of differentiation on chromosome 24. And it turns out that that part of chromosome 24 really is a particular supergene that is common in the southern part of the range. It's pretty rare in the middle and the northern part of the range where these populations were originally from. And what it looks like is that this particular line had a couple of copies of this southern chromosomal segment, and that they were highly selected for in this down line, meaning that they very quickly took over the genetics of that population. And so it serves as experimental evidence that, in fact, these supergenes, when they're passed on all at the same time, can have a strong role in adaptation and can quickly change in populations as selection is working on them. Well, we don't know why these different segments of these minnow chromosomes are acting the way they do, or how these genes are acting. That has to come with very specific research on the individual genes themselves. But it's the patterns that I'm finding interesting here, that the patterns are clumped by chromosome and by chromosomal region, as if these supergenes were common. So where we are in this particular way of looking at it is that we know that natural selection works on genes and that they can evolve. We can follow that process over time, like as climate change happens and ocean warming occurs. But the whole process is also going to be different depending upon the architecture of those differences within genomes. And that architecture is an important part of understanding how organisms are adapting to different environments, and then what their future holds when those environments change.